Hi, this is Christian Brock presenting a talk that I gave to a recent PSI Journal Club session on Bayesian methods for efficacy and safety. The paper I'm presenting is on safety signal detection in ongoing blinded trials. This is the paper here. And this was something I wrote whilst at UCB Pharmaceutical, and the co-authors listed there were all associated with UCB as well, and many of them now remain, but I've since moved on. So ours is a method for Bayesian safety signal detection in ongoing blinded randomized control trials. And that was published at the end of 2022 in Pharmaceutical Statistics. So this talk will be about 20 minutes, and it's going to go like this. First, I'm going to describe why we would monitor safety. Then I'm broadly going to present the problem from a sort of trials and statistics point of view. Then I'm going to talk through briefly our method, and then I'll present some simulation results that demonstrate that our method does something useful. And then I'll wrap up with maybe a thought or two on future research. So why monitor? This is actually a quote from our paper. So we, as sponsors of clinical trials, have a responsibility to minimize risk to participants in clinical studies through safety and monitoring. And now the FDA's final rule for investigative new drugs safety reporting requires routine aggregate safety evaluation, including and I've bolded these for a reason, ongoing and blinded studies. And the reason for the bolding is that just because your trials are ongoing, it doesn't mean that you get out of the obligation to do safety monitoring. You can't just get to the end of a 5-year study and then have your first look at the safety outcomes and then go, oh, geez, we were harming patients the whole time. You need to be doing those interim analyses as you go through to keep patients safe. And also, you don't get to dodge the rules because your trials are blinded. We blind our studies so that we get good, high-quality estimates of treatment effects, and we want to keep them blinded for that reason. But just because your trial is blinded doesn't mean you then have to not worry about monitoring for safety, and that presents a problem. So as sponsors, particularly pharmaceutical companies, will want to keep their trials blinded. But from a sort of patient point of view or from a regulator point of view, there could be a part of your organization that is unblinded, and they could do unblinded analyses. But coming from the pharmaceutical perspective, as I do, we very much wanted to avoid that and use historical information and Bayesian priors to basically let an analysis become tractable without unblinding our study. So this was quite a challenge, but it was one that we hoped we could solve with Bayesian statistics. So the problem, broadly speaking, is that we know how many events there are, but we don't know the arms in which they occurred. That, simply put, is the nub of it. Now let's put some flesh on the bones. Let's imagine we're at an interim, a safety interim. We've got a snapshot of some data. And we know for an adverse event in particular, we've seen 15 events and then we have 60 patients recruited. And we'll also know the randomization ratio and even, so let's say, for argument's sake, it's 1-to-1 randomized and we've blocked it so that we know in those 60 patients, half are in the control arm and half are in the treatment arm. So we've basically got 15 events happened in 30 control patients and 30 treatment patients. Where do those events live? Well, we don't know. One particular arrangement could be that there were 5 events in the control arm and 11 in the treatment arm. Now, in that situation, if you were to place uniform priors, beta 1-1 priors, on the event probabilities in the 2 arms, and then do the sort of posterior calculation of what's the probability that the treatment arm is more toxic than the control arm, given the data, you'll get this little grey box that I've got there, 93%. And that would actually be quite a strong message for a safety signal, and that might have you thinking about what to do next, some kind of escalation to identify what's going on. But of course, in this trial, it remains blinded. Okay, so we don't know that truth was the actual prevailing truth. That's just one of 16 possible truths, actually. If we've got 15 events, they could have been arranged so that there were, like the top left plot shows here 0 in the control arm and all 15 in the treatment arm, and then the posterior probability that the treatment arm is more toxic than the control arm would be overwhelming, and it would pretty much mandate that we did something about it. And then you've got that distribution shifting from treatment to control arms all the way down to the lower right plot. You can see that all 15 are in the control arm and none are in the treatment arm. And then the posterior probability in the grey box of there being a safety signal is virtually 0. So really, there's only 16 possible outcomes here, but which one is the prevailing one really determines if we have anything to do as a sponsor. Now, of course, let's say that we're able to use historical trials and the information and the outcomes contained in those historical trials, so exchangeable populations for our current blinded trial. Let's say that we were able to place a prior on the adverse event probability and effectively the number of events that we'd expect to see in the control arm. And I've represented that here in this bar chart using colors. So the deeper blue in the center represents what we feel like, given our historical data, is the most likely number of events in the control arm. And as we move outwards to the sort of gray tails, those become less and less likely. Now, of course, we know that if we see, say, 3 events in the control arm and there are 15 events overall, then there must be 12 events in the treatment arm, and those would have attached with them similarly likelihoods. And so we get this kind of pairing up, or basically what we're doing is placing a plausibility weight on each of those 16 scenarios. And so when we come to infer what's the probability that there is a safety signal here, we could take, say, a situation like, how do I get rid of that? Oh, for God's sake, go away. We can say, let's take the situation where there's, say, a small number of events in the control arm and relatively many in the treatment arm. And we might say, given our prior information, that this is likely and therefore deserves a high weight. We might look at that situation, the plot's slightly smaller to show it's slightly less plausible. We might look at the situation where there's equal, more or less, events in the control and treatment arms and give that a medium weight. And the little small plot represents it's not likely at all. It is possible that we could see many events in the control arm and not many in the treatment arm, and we would give that a low weight. And what we do is we can weight all of those together to get an overall a posterior set of beliefs. And in this particular example, the numbers are just made up, so that's not attached too much significance to them. But in this particular setting, we've got a posterior probability that there's an 88% chance that the control arm the treatment arm is more toxic than the control arm with respect to this particular adverse event. And depending on the considerations that we would have given to our safety analysis plan at the trial outset, that might or might not meet our requirements to actually escalate and do something about it. Just talking about this big plot in the middle with the densities, you can see that our beliefs on the control arm in the pink color, pinky orangey color, those are they're relatively informative, so the distribution is narrow, and that's because we have lots of historical information. But our beliefs on the current arm, the treated arm, that bluey greeny color in the current trial, they're quite diffuse still. We don't really know anything about those. But what we do know there is that we are sure enough about the red arm to come up with a posterior probability that there actually is greater toxicity in the treated arm. So broadly speaking, our method is that it's a meta-analytical method. We meta-analyze historical trials to derive the predictive distribution from those historical trials. And the predictive distribution is one that's suitable for an unseen trial arm. Okay. We then take the data from our ongoing blinded trial and we create the posterior to infer on the probability that the treatment arm is more toxic than the control arm, given the data and our prior. So this is our meta-analytical model. We start by assuming that the log hazards, and hazards are represented by lambda, we assume that the log hazards are exchangeable across the historical trial arms, and historical trials are indexed by J. And you can see that each log hazard is just estimated by a trial-specific intercept term. Now, if we assume that hazards are constant through time, we can, for a given time t, we can calculate the probability of there being at least an event, and that's by p, and we can rearrange that to get an expression for lambda that's shown there by that fraction. And if we substitute one into the other, we get the fourth expression down there. On the left-hand side, we see the log link function, and on the right-hand side, we see the trial-specific intercept and an offset for the time at risk of patient I in trial J. Now that's an offset term. It effectively has a coefficient of 1. That comes about from the constant hazard assumption. If we were to pop a covariate in front of that log, you would end up with a hazard that changes through time. That's not what we do though. Now, if you were to set time to be 1, so that that means that you're putting all of these historical trials onto some standard time scale, and what we tend to use in our examples at UCB is that t = 1 corresponds to the intended length of follow-up, or time at risk, I should say, in the current blinded trial, so that all of the historical ones are their lengths are expressed on that scale. If we set time t = 1 to be that, then we end up with a probability that we're going to denote by just pi, and the offset term disappears. So now we have the probabilities across the historical trials, pi j, and so this is a hierarchical model, and we attach a super distribution to those pi j, and we use this beta distribution here with parameters shown. What I would say is that the capital N is the aggregate number of patients in the historical trials, and if you work out the effective sample size of that beta distribution there, it's b times n. So b is a scalar on the aggregate historical information, n. And if you were to work out the expected value of that beta distribution, it's a. So a is the kind of a priori event rate in unit time. Now we analyze the historical data, and then we get from that we generate from that a map prior. So here the reddish distribution is the average, or is the distribution of the average event rate in unit time, and you can see that it expects the probability to be about 15% in a year. And we can also generate from that the predictive distribution, which is represented here by the greeny blue color. And you can see that that's more diffuse. That's because it's for an as yet unknown trial. And inherent in this hierarchical method is that trials tend to vary. They vary randomly. So we're less sure about the probability of AE in an unseen control arm. And that's exactly the distribution we would use as our prior on our ongoing blinded trial. But of course, that distribution is just a series of samples that's come out of an MCMC procedure. But what we need to work with for the next stage is an actual analytical distribution. And to get hold of those, we use the RBEST package by Weber et al. And that will generate for you an analytical mixture distribution from your samples. So here you can see we fed in our samples represented by the histogram, and the procedure has come back with a 2-component beta mixture. Those 2 components have weights 64% and 36%, and they have shape parameters as shown there. And as you can see in the plot, the concordance is very good. Now, we then take that mixture prior and use it on the analysis of our ongoing trial data. But of course, now we have an informative prior on the event rate in the control arm, and that means what's left over must be attributable to the experimental arm. And that's how we came up with the 88% estimate that I presented before. Right, implementation. So the Bayesian models were implemented in STAN. We're talking about non-conjugate models here. So I've talked about binary events and beta priors, and you might think beta binomials conjugate, but of course we have this offset term for times at risk. Not every patient has had the standard follow-up time. Some might have been recruited last week, some might have been on for 4 months. There's going to be a difference in the probabilities of having had the event linked to the amount of time you spent at risk. So to adjust for time, we had the model that I presented, and that is what makes it non-conjugate. So had to use STAN to fit the 2 Bayesian models, one model to generate the prior, and the second to analyze the data from the ongoing trial using the prior from the first model. I then wrote helper functions in R, and bundled all of this up into an R package called BSSD for Bayesian safety signal detection. Now that's a package that at the time of presenting, which is November 2023, that package is currently internal only to UCB. But colleagues, when I came up with the package, we always wrote it in a way that we hoped would lend it to being listed on CRAN and published and listed on GitHub so other researchers could use it. Colleagues at UCB, I'm obviously no longer there, but colleagues that remain at UCB are trying to push through right now internal permission to get that listed publicly, and I really hope that happens. Let me show you some results of simulations. So we did a lot of simulations, and there are a lot of axes to talk through here. So first off, these are receiver operator characteristic curves, and the area under the curve shows you the ability of the model as a whole to discriminate situations where there genuinely is a safety signal to when there's not. What we've got here, so the columns of this grid of plots, we've got the historical aggregate sample sizes. So the leftmost is 120. That meant that our historical set was 3 trials of 40 patients each. Then we've got the middle column, which is 300. That was 3 lots of 100 in the historical trials. And on the rightmost column, we've got a large aggregate historical sample size of 1,050. That was actually split over 9 different trials. So going outwards in the columns, we've got different amounts of information in the historical trials. Each of the colors represent the sizes in the ongoing blinded trial. Those go from 50 in red up to 1,000 in purple. The rows of this grid, the top row shows an event rate that's common, that's a 10% risk per annum, and the bottom row shows an event rate that's rare, which is a 2% rate per annum. And we've actually got different lines within the colours representing different amounts of heterogeneity in the historical trials. And those range from none to modest to actually quite a lot. And those actually don't really seem to have a great deal of effect, at least on the ROC statistics. What you do see is that the ROC, and then of course there's the dashed orange line shows a random classifier. So that's one that just randomly 50-50 says whether there's a signal or not. So it has no level of discrimination at all. All of our ROCs are to the top left, so they do discriminate more successfully than random. And you can see the ability to discriminate, as proxied by the area under those curves, increases with sample size. And there are 3 ways that sample size increases here. You've got greater historical sample sizes in the columns. You've got greater sample sizes in the ongoing trials. Those are the colors. And then greater number of events. So the common event, that's the top row, when compared to the bottom row. So lots of information here, but really showing as the sample size increases, the method improves better and performs better. And that's what you'd expect. The one situation where life is very tough is the bottom left, and that's when we have very small, a very rare event and a very small historical sample size. So what this is showing is that having a decent historical sample size is quite important. Next steps. Well, next on the agenda for us so we never investigated Robustification, and after having published the paper, I think that was probably a mistake, and that's something I think we should do now. So we do have that that B parameter, which discounts or upweights the amount of borrowing to do based on it scales the amount of historical information, and we thought that would be enough. But thinking about it, prior data conflict is the type of thing that can happen because our health systems are generally getting better over the world. So outcomes do have a tendency to drift upwards through time. And that makes it entirely likely, perhaps, or increases the likelihood, at least, that the outcomes in the ongoing trial are no longer exchangeable with those from your historical set. What do you do in that situation? Well, one way to deal with it is to have what's called a robust map prior, and that's demonstrated here. So the first component, with weight W, we'd have the beta prior derived from the historical data, and then the leftover weight 1 minus W would just be a diffuse beta prior that essentially admits any probability of adverse event. So in summary, I finish again with a quote. So our method provides a framework to routinely monitor the rate of a selected adverse event, and these are usually serious. Okay, these are normally a problem. These aren't just the types of things we would willingly ignore. It lets us do that in a blinded fashion, and potentially unblind only if the posterior probability exceeds a predefined threshold. Our method will allow us to monitor safety signals in our ongoing blinded studies with the goal of, so why are we doing this? Earlier identification and risk mitigation. So hopefully protecting the integrity of our clinical studies too. So removing that need to unblind until it's absolutely required, if at all. Thank you very much for your attention. My name's Christian Brock. My website's there, and I'm currently at AstraZeneca. Thank you.